Good afternoon. Welcome to our MHRC seminar series this afternoon. It's a great pleasure today for me to introduce our guest, Dr. Walter Herzog, from the Faculty of Kinesiology at the University of Calgary in Alberta. I'll tell you a little bit about Dr. Herzog's background. Dr. Herzog is originally from Switzerland. He did his undergraduate degree in Zurich at the Federal Technical Institute. They're a very famous institute in Zurich. He came to North America and began his education at the University of Iowa, where he did his master's, started his master's, but then fast-tracked to the PhD and was supervised by Dr. James Hay, who is a very famous biomechanist, wrote a textbook that I actually used as an undergraduate and still have it on my shelf. I can see it from here. So trained with him at the University of Iowa, then went to the University of Calgary as a postdoc and stayed there and became an assistant professor in 1990, and he's been there ever since. Dr. Herzog has a large research group, probably 25 trainees, including technical people that help and graduate students, 13 postdocs and graduate students, 4 to 5 undergraduates, total of about 25 trainees. And that helps to explain Dr. Herzog's remarkable productivity. Just check the PubMed listing for Dr. Herzog, and as of today, 501 publications listed on PubMed, 31 alone in 2022 and 4 in 2023. That's not quite one a week, but one every 2 weeks, certainly more than that in 2022. It's just remarkable, and maybe he'll share some of his secrets of success with us today. I'm looking forward to hearing him talk about the role of Titan in muscle function, proposal versus evidence. Dr. Herzog, please turn on your camera and join us, share your slides. It's going to be a treat to hear about Titan. Take it away.
Great. Thank you very much, David, for the nice introduction. I'm just going to share the screen here and see if that all works. Please let me know if you can see my slides and my my pointer as well.
All good. Yep.
Fabulous. Good. So thank you again, David. Pleasure to be here. Talking about my favorite topic, which is muscle contraction, and one of my favorite proteins, which is Titan. So I'll be giving you a little bit background about some of the work that we have been doing and accumulating over the past 20 or so years. And at the end, I'll make a proposal about how we think muscle might be contracting and what Titan's role in this whole mixture is. And I want to say from the very beginning, this is a proposal, although I'm showing some evidence that probably supports this proposal quite nicely. There is really no absolute direct evidence that things happen the way they happen and the way I'm going to be talking to you about. So a little bit of background, it's always been fascinating for me that people have studied muscles for a long time, and it's said that Aristotle, for example, looked at the muscular build of athletes and tried to predict how they would perform in competition. And then, of course, we are familiar with Leonardo da Vinci's beautiful drawings of human muscles. And Alfonso Borelli, for those of you in biomechanics will know that he probably was one of the first ones to try to calculate muscle forces in human movement based on static equilibrium considerations. And then we have the big people of the 20th century in muscle physiology, muscle mechanics, Meyerhoff, Hill, and Andrew Huxley, all of whom won a Nobel Prize. Meyerhoff and Hill directly for muscle contraction, and Andrew Huxley really for nerve conduction, but of course did most of his work on muscle as well. And the other thing that I wanted to emphasize before I start out is that knowledge in science seems to be continuously changing, and the way we think about muscle contraction has changed in my scientific career as well, and it has changed all the time. So if I just go through the 20th century, Hill in the 1920s thought of a muscle as kind of an elastic compound that upon activation would store some potential elastic energy that then could be freed up when a muscle produced work. And of course, this was then proven to be incorrect by his collaborator, Wallace Fenn. Then for a while in the 1930s, we had the idea that lactic acid might be the compound that triggers muscle contraction. But then very beautiful experiments by some people, including Lunsgaard, showed very nicely that when you poison a muscle so that lactic acid cannot be produced, you can still get perfect contractions in a muscle. So that kind of got rid of that particular theory. And then, you know, before the 1950s, between the 1930s and 1950s, a lot of people thought about muscle contraction being caused by the shortening of the myosin molecules, the A band in the sarcomere, that then would produce shortening of the muscle and force production. And again, today, obviously, we think that's probably not quite correct. So then one of these paradigm shifts that has held up to this day is really the sliding filament theory, when Hugh Huxley and Andrew Huxley in their May 22nd Nature publications proposed that maybe we have these two sets of filaments, actin and myosin, that slide relative to one another. And then Andrew Huxley in 1957 proposed a mathematical framework and also a biological framework on how this sliding of the actin versus the myosin filament could be occurring, which we now refer to as the cross-bridge theory. And so today, in my talk, I'll talk a little bit about what I call the old paradigm, kind of the cross-bridge theory that you see in your muscle physiology textbooks. And then I talk about what I would call present observations. These are really observations over the past 20 years that we have made, and they are focused on the work that we have done. Other people have done absolutely excellent work as well, but I'm really focusing on some of the work that we have done in this particular talk. And then, as I said, I'll give a little bit of proposal of how I think how actin, myosin, and titin might be producing work and why titin might potentially be playing a role here. So if you start out with the old paradigm, so Andrew Huxley's 1957 theory and all the adaptations that it underwent. So just for a little bit of a review of the mechanics of that, this is really textbook knowledge. We know, you know, that a muscle is composed of some subunits, fascicles, fibers, myofibrils, and we work a lot on this level, on the myofibrillar level, which is an organelle that has sarcomeres that are serially arranged. So it's just one sarcomere after the other. There's nothing in parallel to it. And the other good thing about it is that you strip it of all really the passive structural elements, except some very few that I will be talking about in a second. And then, of course, we have a single sarcomere that you typically see with the myosin filament here in the middle, and then the thin actin filament at the end going into the Z line. And the idea is that the thick filament or the myosin filament is built up of these myosin molecules that has this tail portion, and the tail portion is incorporated into the thick filament. And then you have these two globular heads that are sticking out from the backbone of the thick filament. And these are now called cross bridges. And we think that they interact with actin and pull the actin past the myosin filament. And then the actin filament is made up of these two filamentous chains of actin globules that are wound around each other. And then you have in this filamentous groove, you have the tropomyosin, a regulatory protein, and then you have the troponin C, the troponin complex with troponin C that binds calcium and then is thought to undergo configurational changes, these regulatory proteins that then allow for cross-bridge attachment. And so that's how it all works. And so today we think about it in this way, that we have the myosin molecule here, we have an elastic element that connects to these two cross bridges. A cross bridge will attach to the actin filament, will undergo a rotation of about 45 degrees. And then by doing that, will pull the actin past the myosin filament. And this is all combined. All these mechanical steps are nicely combined with the hydrolysis of ATP. And in a traditional theory, it's thought that we have one ATP per cross-bridge cycle. So this is how we really think about it. And this cross-bridge theory is, mathematically speaking, when you take it and you make mathematical predictions of it, it's typically very good for predicting force and stiffness and the energetics of muscle contraction, as long as you work with isometric or concentric contractions. That means contractions when the muscle length either stays constant or when the muscle shortens. However, and Huxley in 1957 already admits that in eccentric contraction, there is a lot of things that don't really quite work. So for example, in the 1957 model, the force, stiffness, and energetics in the cross-bridge model was vastly too high. There's too much energy required, too much force, too much stiffness. And so when you then try to fit the eccentric contractions, then the isometric and concentric contractions don't fit anymore, and vice versa. And so there's something happening in eccentric contractions that in the traditional classic model of the cross-bridge theory is not really well explained. And Huxley recognized that. I mentioned already in 1957, he spends quite some time on explaining why maybe eccentric contractions could not work. And then in his beautiful book that Andrew Huxley wrote in 1980 called Reflections on Muscles, he talks about eccentric contractions. And one of the things he says is that there is this large stretch that is applied to muscles in eccentric contraction, and it leaves the tension raised for a very long period of time. And he then says, and we cannot really predict this. But then in the proof for the book, there was this interesting footnote added. So this footnote then says this raised tension, the one that he talks about above, is a consequence of non-uniform elongation of sarcomeres during stretch, and therefore it does not require an explanation in terms of the cross-bridge behavior. And a lot of people then at that time thought that many of what I call these history-dependent effects, or this increased tension that you get in a muscle following stretch, could be explained by this behavior of non-uniform sarcomeres. And I'll be talking about that right now and next. So we're going to the present observations, and I want to introduce this idea of the increase in force that you get after a muscle is actively stretched. What did Huxley mean by that? And what do we mean today by that? So imagine this is work that we did on a single fiber, intact fiber, where you have an isometric contraction, the length doesn't change, and you just activate the muscle and you get a nice steady force, maybe with a little bit of a decrease because of fatigue, but essentially a nice steady state force. And now you take that same fiber at a shorter length and you stretch it actively, then of course you get this little bump here that's predicted based on Hill's force-velocity relationship and also partly by the cross-bridge theory. But then the force stays a bit higher than in the isometric contraction, despite the fact that you're exactly at the same length. It's an isometric contraction and you have the same activation level. And if you go to a shorter length and stretch it a little bit more, then you get even a higher force. Or if you do the opposite experiment and you shorten the fiber from a longer length to that isometric length and let it sit there for several seconds, then you get less force. And this is really the puzzling thing that cannot be explained by the cross-bridge theory, because in the cross-bridge theory, after 500 milliseconds or a second or so, when you have reached a steady state and you're at the same length, the same activation, isometric, everything is the same, it will predict that it gives you the same force, the force that corresponds to the isometric force. But that doesn't seem to be happening. And so today I want to focus mostly on this force enhancement, not on this force depression. And so how can we potentially explain that? And as I mentioned, the cross-bridge theory would essentially predict that you don't keep this enhanced force, but the cross-bridge theory will predict something like that. Yes, you get this transient increase in force during the stretch, but then that subsides and it falls nicely together with the isometric contraction. And so the idea now is, how did this happen? And I mentioned that Huxley and other people, Hill, Paul Edman, Uwe Proske, Dave Morgan, all the leading muscle physiologists of the 20th century didn't really think much about this because they were very happy to think that sarcomeres are unstable on the descending limb of the force-length relationship, which is this part of the force-length relationship where we see this behavior, and that we can explain this all with sarcomere length non-uniformity that occurs when you stretch a muscle. So how does this look like theoretically? So the idea is that you have a passive muscle, you stretch it to a certain length, and you activate it on this descending limb of the force-length relationship, and you get a certain amount of force as predicted by the amount of overlap of actin and myosin. And then you take this fiber or this muscle, you deactivate it, you stretch it a little bit, you wait a couple of minutes till it's nice and fresh again, and you activate it. And since now you have less overlap, you get less force. And that is all very nicely predicted. And if you do the experiment in this particular way, that's exactly what you're getting. So the question now becomes, what happens if you don't deactivate the muscle, but you leave the muscle activated and you stretch it from here to there? So according to the non-uniformity, sarcomere length non-uniformity theory, the idea is that most of the sarcomeres in a stretch will not really elongate very much, and they stay much higher up on the descending limb than where the average force would be if all sarcomeres stretched the same amount. However, in order to get this average sarcomere length, you have then some sarcomeres that are stretched really a lot, and they are going beyond actin-myosin filament overlap, and they're caught by some passive force until they are roughly in a force equilibrium with the active sarcomeres. And therefore now you get a force, that was the idea, you get a force that's this high, but the average sarcomere length is this. And if you isometrically activate the muscle, you would get this amount of force. So here is the extra amount that you're getting. There's a couple of things that have been known for a long time that are really puzzling about this particular theory. And one of the puzzling facts is that as early as 1940s and 1950s with Ramsey and Street and Huxley and Peachy and other people, they have already documented very, very nicely that in vertebrate skeletal muscles, you always have non-uniform sarcomeres. You never get in an isometric contractions uniform sarcomeres. And the whole premise that an isometric contraction is executed and performed with perfectly or nicely uniform sarcomeres, that has been known to not be correct. And then there's a lot of other predictions that you can make based on the sarcomere length non-uniformity theory. And we tested many, many of them. And I'm just going to show you two that I think are the most convincing that this is probably not a feasible mechanism. The first one is that when you look at the mathematics of the sarcomere length non-uniformity theory, then the maximum force that you can obtain is when the active sarcomeres are on the plateau here of the force length relationship, where they can exert a maximum force. And then you have other ones, here on the passive limb. But this is the force that you can get. So based on that theory, the sarcomere length non-uniformity theory, you should never get forces that are higher than the plateau of the isometric force length relationship. And we did experiments. This is again single intact muscle fibers where we got the sarcomere force length relationship, this nice isometric contractions, and it follows very nicely what the theory would be predicting based on filament overlap. However, if you now stretch these intact fibers by a certain amount from certain different distances and different origins, then you will see that some of these steady state forces at the end of the stretch, these force enhanced forces, are clearly 10, 15, 20% above the plateau that a muscle or a fiber is able to reach isometrically. And so therefore this extra force for these sarcomeres that are here needs to be explained. So the idea is something else has to happen. And so therefore that cannot really be predicted with this particular theory. And then I think really the most compelling evidence was when my PhD student at the time, Tim Leonard, for the first time isolated mechanically a single sarcomere. So here you see a myofibril between our micro electronic machine force levers. And then we have two motors here, and this is controlled manually. And we try to isolate this single sarcomere here. So here we have the four dark bands, the I bands, that's a sarcomere. And then you have a sarcomere attached here. And so now Tim tries to go to this Z line here to mechanically isolate this single sarcomere. And it looks all really nice and easy, but it took us about a couple of years to really prepare this perfectly and, and make it happen that we that we could do this isolation of the sarcomeres. And because it takes such a long time, nobody except for trainees of mine have ever done this. And some of them are now at different universities, but when you see a single sarcomere work, mechanical work, that those are, so as far as I know, all still people from our group that learned it here in Calgary. But anyway, so then we did these experiments with a single mechanically isolated sarcomere, where we do an isometric contraction at some long lengths, and we get the force, the steady state force. And then we take the single sarcomere at a shorter length and we stretch it and then see what the force is. And you can see here, we stretch it, and you get a very similar behavior as you would get in a fiber or in a muscle. And then you get this decrease after the stretch of force. But this steady state force that lasts for a very long time is much higher than the steady state force that you get in the isometric contractions. We have an enormous amount of force enhancement in a single myofibril. And you're asking yourself, how can they hold this for 20 seconds without fatigue? And of course, when you do the calcium handling in your solution, then the muscle actually doesn't get fatigued, and you can hold this for a minute or two without any decrease in force. So if you then do this with multiple isolated sarcomere, you normalize it all to the isometric force on the plateau, and then you measure the isometric force on the descending limb, and you get pretty much what you would expect, a slightly higher on average, presumably because there's already a little bit passive force there. And then you look at the forces that you obtain after active stretching of the single sarcomere, and this indicates the enhanced force, the enhanced steady state force that you get exactly at the same length, here for an isometric contraction, and here for an isometric contraction that was preceded by a stretch from here to there. So an enormous amount of force enhancement. So really the simple conclusion was that sarcomere, the force enhancement property seems to be contained in a single sarcomere, and I don't need many sarcomeres that are non-uniform in lengths relative to one another in order to get this property. So sarcomere length non-uniformity likely is not really a factor that is required for getting this additional force, this force enhancement property that I have been introducing earlier. So then the question becomes, okay, so now you say that's not correct. So what's happening now? What's the alternative? What might be happening here? And that's where I'm going to talk a little bit about a new paradigm or a new way of thinking about it, and experiments that we did that might support what we are trying to say, and maybe you find some flaws in the arguments, and I would be very, very happy to hear about those. And I always tell people, a few times in my scientific life, 3 or 4 times, I did an experiment, and something happened that I wasn't looking for at all, and it changed completely my mind. And one of these experiments I did on cat soleus preparation in 2002. Tim Leonard and I did the experiments by ourselves, and it changed our mind. It changed a lot of people's mind in the biophysics and the biomechanics community. And the simple experiment we did is we took a cat soleus muscle in a live animal, and we activated it, and we get a certain amount of force. And this is at a very long length, where we also get some natural passive force. And the idea of this study was we just wanted to see how force enhancement depended on the length of the muscle. So then we stretch the muscle passively as well from a short to a long length, and we get about the same amount of passive force. And then we stretched it actively, so we activated the muscle. We get this hill bump here when we stretch it, and we get the force enhancement as we expected. But then we deactivated the muscle, and so now it's a passive muscle. And in many of these experiments, the passive force now after the active stretch stayed higher. And initially we thought, oh, we are stretching it not to the right lengths, we are at a longer length here or so. And then of course you do all the control experiments, and you realize that no, very often you will get this additional passive force after an actively stretched muscle that you do not get if you just activate the muscle here in white, or if you just pull it to a longer length passively. But if you pull it to a longer length and it's activated, then you get this passive force. And so we published this in the Journal of Experimental Biology, called it passive force enhancement. And now if you go into the literature and look at passive force enhancement, there is dozens and dozens of papers from our group and many other groups coming up that now talk about this property. So then we said, if there is really more passive force that somehow comes about when you actively stretch a muscle, where does it come from? So we did experiments on different structural levels. And one of the levels was the single sarcomere and the single fiber. And to make a long story short, we found this extra passive force after an active stretch in myofibrils and sarcomeres as well, compared to when we just pull the muscle passively. So here we have sarcomeres that are pulled from 2.4 to 3.4 microns on average, and the passive force after an active stretch remains higher than after a passive stretch. And then you ask yourself, well, why is this so important? Because you've already shown in the muscle. The important thing for us was that in a single sarcomere in myofibril, the only passive force that you have, or I should say 99% of the passive force, seems to come from titin, the structural protein. If you eliminate titin, you don't get any passive force in a myofibril. And so we therefore said, well, likely this is from titin. And then the question becomes, well, what is titin? Here we have the myosin that you're familiar with in the middle of the sarcomere. Here you have the actin. And then titin is the structural protein that goes from the Z band, runs freely like an elastic spring that can be extended and shortened when a muscle or a sarcomere elongates or shortened here. And then here in the A band, it is attached rigidly to the myosin filament, so there's no elongation here. And then it terminates in the middle of the M line. And then you have a 180-degree opposite directed titin here along this myosin filament going to this Z line here. So it spans a half sarcomere, and it has this property where it's viscoelastic here, and it can stretch to very long lengths, and it can refold. So you have this spring-like behavior. And so we said, well, titin is the only thing that can produce passive force. It seems to behave differently when we activate a muscle and we stretch it, so we get more force. So the easiest way to think about this is if this spring here of titin is adjustable and it becomes stiffer when a muscle is activated compared to when it's passive, or in other words, you get more force from this passive structural element when it's stretched in an active state compared to a passive state. So really the hypothesis then was that we put forward in 2002 is that titin is a calcium-dependent molecular spring. And we presented that as an abstract and a poster at the Biophysical Society conference. It was really interesting because half the people thought this is a really super great idea, and half the people thought, what nonsense, this is just not going to happen. And so we felt, well, big controversy, let's see what's happening. So the idea was that we have titin here in a short sarcomere, and we stretch titin passively. And when we do that, we know we get a certain amount of force. So we knew that. So now the idea was, what about if we add calcium? So we activate the myofibril with calcium, and we see if we then get more force from titin, presumably because maybe calcium might be binding to these molecular bonds and might reinforce them, and therefore might make titin stiffer. And therefore, if calcium is in the mixture, we might get a different behavior, a different mechanical behavior of titin. So we did that, or we tried to do that. So here's a myofibril, and we wanted to stretch this myofibril in a low concentration of calcium that corresponds to a resting level, and one that's high that corresponds to a saturating level. The problem, of course, is when you do that in a myofibril and you expose it to calcium, actin myosin cross bridges will interact, and then you cannot decide what's the active force that comes from myosin cross bridges and what's the passive force from titin. And so there's various tricks. You can inhibit cross-bridge attachment chemically, and we use, for example, butanedione monoxide, or you can also delete troponin C from your myofibrillar preparation. That's what we did in this particular experiment that I'm going to show you. So from the troponin complex, we took the troponin C out. Therefore, calcium cannot attach to this complex anymore. Therefore, when you activate a troponin C deleted myofibril, you don't get any force. And of course, we've verified that you don't get any active force. And we also, through the electrophoresis, verified that the troponin C complex was indeed deleted. And so now we can stretch this myofibril in these various different calcium concentrations. And as expected, when we stretch it by the same amount, we get less force in the low calcium concentration, and we get more force in the higher calcium concentration. So we were very happy about that. The next question that we had then is, where might this calcium be binding and what might it reinforce? And we worked with rabbit psoas myofibrils mostly, which is shown here. And you see that it has a lot of these brown rectangles. And these brown rectangles are called immunoglobulin domains. And they are known to unfold when there is enough force there by quite a big degree. They go from about 4 nanometers. And when you break 2 molecular bonds, they go from the 4 nanometers to about 28 nanometers, and you have lots of them. So we figured that might be a good target. So therefore, we then recombinantly constructed 8 immunoglobulin domains in a row. And then we wanted to see if calcium indeed attaches to these immunoglobulin domains, and we used fluorescence spectroscopy for doing that. And then we thought if that happens, then we also wanted to see whether or not it makes a mechanical difference, and then would use atomic force microscopy to see, does it affect the mechanics? And so initially here we measured the fluorescence intensity of these I-27 domains, that immunoglobulin domains, in the absence of calcium, with a little bit of calcium, with a little bit more of calcium, and you can see that the fluorescence intensity is depressed in a dose-dependent manner, indicating that if you have more calcium is binding. And then if we take the calcium away, then this goes back to the initial level. So we have a dose-dependent reaction or binding of calcium to these immunoglobulin domains that's reversible. And that's exactly what we were looking for. And so then we attached these strings of immunoglobulin domains to an atomic force microscope, and we pulled these, and you get this very funny pattern when you pull immunoglobulin domains and nothing else, because the force gets higher, and then all of a sudden, one of these immunoglobulin domains will unfold, go from about 4 to about 28 nanometers, as I mentioned before, and then it becomes slack and force decreases, and then you go to the next one, and to the next one, and so on. And so we did this under controlled situations with low calcium and then in saturating calcium. And then we did this literally hundreds of times with different immunoglobulin domains, and then found that in the controlled situation, we got a certain amount of force that was required for unfolding these immunoglobulin domains, around here at the beginning, about 180 piconewtons. And in the calcium saturated preparation, it was a bit higher. It was about 20% higher. And that was very consistent. You can say, where are your standard deviation or standard error bars? They are actually here, and some of them are smaller than the symbol, and that's why you don't see it. So you don't need to do any statistics. This is highly significantly different. It takes more force to unfold these immunoglobulin domains, or that particular immunoglobulin domain, in the presence of calcium. And so we showed that. So what I showed you here was this particular study by Mike Duval that was published in 2013. In our group, we also showed that the glutamate-rich region, the E-rich region in another domain of Titan, which is called the PEVK region, also binds calcium. It also increases the stiffness. And Siegfried Labeit and Henk Ronsier showed that actually already earlier that this is indeed happening. So we are fairly certain that calcium can and will bind to Titan, and that it affects the stiffness of Titan. So that was all good. The only problem was when we then did this with just calcium activation, the increase in force that we got was very small. It was only about 10% of what was required to explain the passive force enhancement or the residual force enhancement. And so we figured, if this is really Titan, something quite different must be happening. And so then the next idea we had was the following. We said, well, maybe Titan doesn't only get stiffer by changing its inherent stiffness by calcium binding, but maybe Titan can make itself shorter, so that only part of the free spring would be available to elongate, and the other part would not be available. And then, of course, when you make a long spring short, but you stretch it by the same absolute amount, then it would be much stiffer and produce much more force. And then you ask yourself, why would we have such a crazy idea? The idea really was because we know that titin naturally binds to actin at the very, very end here, just about 50 nanometers before it enters the Z line. We also know that we have 6 titins per half myosin, and one of the titins goes exactly to one actin. And so it seemed feasible to us that maybe some binding might occur. And then we also know that in some invertebrate muscles, you have titin analogs. For example, in limulus muscle, you have a twitchin, a protein called twitchin, which is a precursor of titin, kind of an evolutionary precursor. And we know that twitchin can make bonds based on phosphorylation between myosin and actin. And so we figured this might not be really so unreasonable. So the idea then was, okay, let's test check if we can do that. So the idea was that we put markers, fluorescent antibody markers on the titin element here, and we measured segmental elongations of titin when we stretch it passively, as here, and then all these markers, all these segmental elongations would be, all segments of titin would be elongating, or we would do it actively in the active state, and then maybe some of these segments, and we were speculating more the proximal segments, where you already have here a natural attachment, would not elongate, and this element here, the distal element, would then take up the entire elongation of the muscle or of the sarcomere. And so we did this. So here we see some labeling. The label that I'm going to show here is called the P146, and it labels, for those of you who know titin, at the distal end of the PEVK region. And so we have this marker now that I'm showing here schematically. So imagine this is the Z line, this is titin, this is actin, this is myosin. So imagine that we have this marker here somewhere a little bit beyond the middle of titin, and we measure now this distance from the Z line to this fluorescent marker, and we stretch this passively, and we also measure the elongation of the half sarcomere, which is already given here. And so here we have the first data point of this distance from the Z line to the marker. And in the passive stretching, we would expect that this distance increases. And indeed, as shown here with the actual data, it increases. And then if you watch now the animation, so we imagine that this is what's happening. Yes, it elongates. And since this elongation is not as much, this slope is not as steep as this one, we also knew that there must be some other elongation. So therefore, this part here would have to elongate as well in order to explain that. So the idea is in passive, both elements elongate. So now in the active stretching, we thought that this marker now, or around this marker, this will be binding to the actin filament upon activation, and therefore this marker would not be moving, and all the elongation would be taken up just by this distal segment. So this marker would be staying nice and horizontal, and that did not happen. What happened in about 120 sarcomeres where we measured this is that we always had some increase in this distance, and then it became flat. So the interpretation initially was that maybe what happened is that this is increasing, and then the marker, where the marker is, it attaches to the actin molecule, and therefore doesn't go any further. Of course, so that's the interpretation there once more. But of course, another interpretation could be that the attachment does not occur where the marker is, but maybe more distal to the marker. Maybe it would attach here and not at the distal end of the PVK region as we speculated. And therefore, you would have an initial elongation here that then gives you the remnant elongation that can be accomplished here. But then since this spring is now shortened, it would run out of elongation. It would become very stiff and then couldn't elongate anymore. And so schematically, the way you might imagine this is like this, that titin is attached here already to the actin, but not where the marker is, but a little bit more proximal. And therefore, you have this element that can now elongate, but then it's stretched to its total length and cannot elongate anymore. And so it doesn't get longer anymore now, not because it's attaching here, but because it has stretched out the free spring length. So once more here, if you show this in once, so this stretching out, and then the marker stops because this is coming to its end length, and then the rest has to be taken up here. So there's probably different interpretations of this as well, but that's how kind of we thought about it. The one thing that has been very consistent is that at one point in longer stretches, this element, this proximal element, does not elongate any further. It seems to get stuck, and then all the elongation has to be taken up with the distal part of the titin molecule. So I'm slowly coming to the end here. So what I'm trying to tell you today is that we think that titin is a soft molecular spring in passive muscle stretching, and we know it is. You can test that in muscle, in myofibrils, in fibers. There is not much resistance of titin in a muscle, in a passive muscle. But we think that titin might be stiffening itself, and therefore when you actively stretch a muscle in an eccentric contraction, that you get much more force from titin than you would in the passive scenario. And the idea is that maybe calcium binds to titin, thereby changing its stiffness. And I think this is fairly accepted and has been shown by different groups, not only by us. So I think this is fairly accepted that this is happening, but it doesn't seem to have great functional relevance. It doesn't seem to be a big effect that you get with that. And then the other idea is that titin might be binding to actin. We saw that this proximal elongation in the active muscle seems to be stopped. But we don't know, of course, if titin is really indeed binding to actin, or if something else might be happening there that does that. But in conclusion, really, we speculate, we propose that force regulation in muscle does not only depend on actin and myosin, but that titin appears to play a crucial role in this interaction as well. And I used to say a couple of years ago and that this was a neglected role, but that's really not true anymore in the biophysical community. That titin plays some role in regulating contraction, and that it somehow interacts with actin and myosin, and that it behaves differently in active and passive contraction. That is fairly well acknowledged now, although how this exactly works is still very, very much a matter of debate. And so when I talk about a paradigm or a proposal, how does this work now? So we modeled this theoretically with a mathematical colleague of mine, Gudrun Schappacher from Austria. And really what we did in order to model all that and make good predictions in eccentric contraction is as follows. We leave the cross-bridge theory and the isometric force-length relationship of actin and myosin absolutely normal as it is. And the passive force in a passive muscle is also absolutely normal as shown here. But the proposal now is when you activate the muscle, two things happen. This passive force that's based on titin, and of course there's other elements that contribute to passive force, but in a sarcomere or in myofibril, where you only have titin, the following will likely happen. First of all, titin seems to shorten itself, and therefore the passive force occurs at a shorter sarcomere or fiber muscle length than in the passive muscle, and the increase in force with a given amount of stretch is steeper because there is more stiffness. So when you activate a muscle, we propose passive force from titin comes in earlier and it is stiffer. So the idea being calcium binds and titin somehow makes itself shorter, possibly by attaching to actin in this particular way. So I want to finish up my talk by coming back to Andrew Huxley. I've met him on several occasions. The last time in 1999, when he came to a muscle meeting in Calgary, and these are my two oldest boys. They were 7 and 9 at the time. Sorry, 5 and 7 at the time in 1999. And here's Andrew Huxley when we went to the Drumheller Museum. And I want to bring up a quote that he had also in his book from 1980, where he talked about eccentric contractions, and he said something like, I imagine that special features have evolved which allow elongation to take place without damaging the muscle. So he talked about eccentric contractions and felt that something is missing here. There must be some special feature that allows us to explain why muscle doesn't get damaged, why sarcomeres don't pull themselves apart beyond actin myosin filament overlap. But of course, in 1980, titin had not really been discovered. I mean, Maruyama suggested, discovered Titan in 1976, and then Wang came in 1979. So when this book was written, really we didn't know anything about any functional importance potentially about Titan, except maybe that it produces some passive force and maybe tries to center the myosin in the middle of the sarcomere. And so I think, Andrew Huxley in his genius foresaw that there might be a special feature that becomes particularly relevant in eccentric contraction. And I think he anticipated something like Titan to come onto the place. So with this, I would like to thank David once more very, very much for the invitation. And I'd be very happy to answer any questions that you might have.
Wonderful, wonderful. That's wonderful. Thank you so much, Walter. That's a great, great presentation. I've got a page of notes here that I've taken down. Folks, we will take questions through the Q&A feature on this. So I'll just give you a second to type your questions in, if you don't mind, and I'll maybe start the questioning by asking you about. So Titan clearly has a protective effect, preserving sarcomere integrity during an eccentric contraction. Is that a fair statement to make? Because the sarcomere is actually trying to cross bridges are trying to shorten the sarcomere, and yet the muscle is lengthening, and Titan is, so the sarcomere is lengthening, but Titan is preventing it from falling apart. Is that pretty simple, but is that accurate?
Yeah, I would say that's accurate. And, and people who modeled the cross-bridge theory just with actin and myosin filaments, they either had an unstable behavior mathematically, or they had to somehow artificially make sure that the myosin filament remained in the middle of the sarcomere. So probably the most important function of Titan that I really didn't talk about is really to keep the myosin filament roughly centered in the middle of the sarcomere, because due to the stochastic interaction of cross bridges on one half of the myosin and the other half of myosin, if that was not the case, you would indeed have instability, and the myosin would be pulled to one side of the sarcomere all the time.
Right. Now, you didn't talk about the heart at all, but I wondered if the stiffness of Titan had a role to play in keeping heart sarcomeres on the ascending limb of the length tension curve. Would that also be true, or what do you think about the heart?
Yeah, no, that is also correct. We have done some cardiac work, little, but other people have done more. Henk Gronzier, Wolfgang Linke did a lot of work on the heart, and as you may know, the heart, operates in a mouse and in a rat at least over about 1.8 to 2.2 microns sarcomere lengths average.
Yeah.
But at 2.2 microns of sarcomere lengths, when you reach the end of the plateau, the passive force is almost as high or as high as the active force.
Right.
So the passive force in a cardiac myofibril and cardiac muscle is much, much greater than what you would expect in a skeletal muscle. And that has to do with the different isoforms. I didn't talk about that, but every muscle seems to have a different Titan isoforms and a different lengths. And in cardiac muscle, the Titan is much shorter than it is in all skeletal muscles that I know, and therefore is much stiffer and produces much more passive force at much shorter sarcomere lengths than what you would ever see in a skeletal muscle. That's absolutely correct.
Right. Okay. Let's take some questions from the Q&A. I'll read them out.
Sure.
Matt Russell says, thank you very much for the talk, Dr. Herzog. Doesn't ask a question. Someone's asking, will the session be recorded? And it is being recorded. And with Dr. Herzog's permission, we will post it up on YouTube eventually. You okay with
Absolutely.
that?
Absolutely.
That's
yes.
Fantastic. Eric Jacques, wonderful story. Thank you for sharing. Have you tried throwing Titan and actin in tools like AlphaFold to get an idea whether interactions between these two proteins are computationally predicted and where that interaction is working?
That's a great question. And we have not done interaction experiments, but other people have. So again, I go back to Wolfgang Linke, who did motility assays. So motility assays where on a cover slip you have like a lawn of myosin cross bridges that reach up, and then you have actin on top of it, and then actin is propelled forward, and you can measure the speed as a function of ATP concentration. And then if you add certain segments of titin to that motility assay, then people have shown that the actin speed with which it's propelled is much slower. And the interpretation has been that, yes, there is indeed interaction between some of the titin segments and actin that slows it down, and therefore that happens. We have done some work on mouse muscle where we eliminated some very specific segments of the titin protein, and one that we had was where we eliminated a segment that's referred to as the N2A segment. And when we take that N2A segment out, then this radically increased force in a stretched myofibril and sarcomere, we don't see that. And so this result would suggest that the N2A region is crucial for the binding of titin to actin, always assuming that titin to actin binding actually occurs. But when you eliminate that particular segment, then the property of force enhancement and passive force enhancement that you just showed, it's still there, but to a much lesser degree in that particular mouse model. Okay. So there is some evidence there that, yes, there is some interaction between titin and actin in other preparations than just the way we've done it.
Okay. Matt Russell asks if you have looked at different levels of contraction intensity or different levels of calcium availability in the sarcomere and investigated if this changed the actin-titin binding response to bind sooner or later during active stretch.
So we have done in myofibrils and in skin fibers, we have done these experiments at different levels of calcium exposure. So, you know, we measured the calcium sensitivity of a fiber of a myofibril, and then did experiments at levels where the force production was reduced due to the reduction in the calcium concentration. And when you do that, it appears that the absolute value of the force enhancement stays about the same, but the relative value, because we usually express it in % of the isometric reference contraction. And since the isometric reference contraction has a smaller force when you reduce the calcium concentration, then you have the same difference, the same absolute difference, but you divide by a smaller reference value. And therefore, it usually turns out that in submaximal activation, the relative force enhancement is bigger than in a fully activated myofibril or fiber or muscle. But not because you get more, but because your reference contraction has less force and you divide by a smaller value.
Okay, yep. Dr. Stuart Phillips says, fascinating presentation, Dr. Herzog. I know it's not titin related, but maybe it is. Are sarcomeres the smallest independent contractile unit in muscle?
Ooh, that's a tricky question, and most people probably would say yes. I think it is not. And when I say independent unit, is sarcomere the smallest independent unit? When you look at people who model single myofibrils like Ken Campbell and Yachin Denot and other people, they think of a sarcomere, you know, this is a Z band, this is a Z band, that's where it stops and finishes. And then you say, okay, this sarcomere produces a certain amount of force, and in order for the Z line to stay where it is, the sarcomere on the other side has to exert the same amount of force. So you think of the sarcomere, the Z line, as a wall with sarcomeres independently on one side and the other side pulling on that wall. I don't think about it that way because I believe there is cross connections across the A band. And so if you think about, is a sarcomere, mathematically speaking, an independent contractile unit? And I would argue, no, it's not. And therefore I would argue, no, a sarcomere is not the smallest. It's the smallest maybe contractile unit, but not the smallest independent contractile unit. I think it's very much affected what happens in the next and the next and the next sarcomere.
Right. So his second part of the question is, what are the true series elastic elements of muscle, and what is the exact role of aponeuroses? Like I thought you were talking titin is the series elastic component here, and he's asking the same question. And what is an aponeurosis, and how does it fit into that series elastic function?
Oh, you're getting into big trouble here. Let me start with titin. So in a passive muscle, if you assume for the moment that there's no cross bridges attached, even in the weakly bound state, so actin and myosin are not interacting at all. If you then think of a sarcomere and you pull it passively, then I would argue that the titin is in series with the myosin. Or in other words, if you measure the force, if you cut the myosin and put a force transducer in there, and you cut the titin, you put a force transducer in there, you would measure the same amount of force because they're strictly in series. But as soon as you activate the muscle and you have cross-bridge attachments, then that's clearly not the case anymore, because titin force will then be determined by how long titin is, and it's completely independent of the force that goes through the myosin filament, because then all of a sudden the titin that connects to the myosin and at the very end to the actin becomes in parallel with the cross bridges, and therefore, mathematically speaking, is clearly not in series anymore. So my answer always is titin is ideally, in an idealized passive muscle, is in series with myosin, but in an activated muscle, it's clearly in parallel. So the titin force adds to the force that is produced by the cross bridges. Then in a whole muscle, I would argue, well, it's fairly simple to argue that a tendon, an external tendon, is in series with the muscle. But an aponeurosis, I'm absolutely convinced, is not. And I know a lot of people have made measurements of elongation of aponeurosis and estimated muscle forces, and then they calculated stiffness or moduli. I personally think, and I think we have some very, very good evidence that that is not correct. Because to me, coming more from a mechanical background, to me, when you say something is in series, that means you have two springs in series. That means the force in this spring and in this spring is the same or proportional at any given instant in time. However, we know in an aponeurosis that's not the case. Aponeuroses are quick at one end, and then they fizzle out. So just from the anatomy, I would argue you can see that the force in an aponeurosis actually continuously changes and is not the same as in the tendon. And you can also show that mathematically. And then we have made measurements where we, in cat medial gastrocnemius and recently in sheep medial gastrocnemius, where we measure the aponeurosis lengths as a function of the directly measured muscle force. And interestingly, what we see in the stance phase, when force goes up like crazy and comes down, so big changes in force, the aponeurosis stays almost at a constant length. And then when you have no force in the muscle in the stance phase, but the muscle undergoes a big length change, then you get big length changes in the aponeurosis as well. So you have lots of length change when there is no force acting on the muscle and virtually no length change. And so I think there's really compelling evidence that aponeuroses are not in series. And when I say not in series, please understand this as from a mechanical engineering point of view. I mean, structurally, you can say, yeah, an aponeurosis is in series with the tendon because it connects to the tendon and, and structurally goes there. But if you talk about stiffness, if you talk about elastic modulus, if you talk about energy storage, and you talk about mathematical terms, then people have treated it as in series, and I think that's completely incorrect.
Okay. Well, your talk has generated a lot of questions. Are you okay for a few more? Because it's now Are you okay?
I have a little bit more time. Yep.
Okay, a few more questions then. Jeff Power says, great talk. What are your thoughts on the role of myosin acting as a mechanosensing structure, mobilizing additional myosin heads, putting in the on state during high force eccentric contractions? You think that has any role in residual force enhancement?
So, oh, sorry.
I was going to say, I can repeat that if you want.
No, I understand. I know exactly what the question is, because it's a very good question, and people are working on that right now. And I can only say what I'm thinking about it, because I have not made direct measurements of on and off states. We have been to the synchrotron, the X-ray, the low angle X-ray diffraction facility in Chicago, but we have never really specifically looked at that. But from what I know and what I've read, I think it's a very appealing idea that cross bridges have different rate functions of attachment and detachment. And I have seen X-ray diffraction results from other people that very strongly suggest that we have this off state and this on state of cross bridges. So, without having done the experiments myself, it's a very appealing and intuitively appealing theory to me. And in fact, I have published a little paper on the mathematics of all that and how it potentially could work. So I believe that. The next question then is, could that potentially influence force enhancement? There, I would say my argument would be probably no, because if the argument is that with the stretch we recruit more cross pitches in the on state, and therefore we would have more cross pitches attached in the force enhanced state, there's a lot of evidence that that's probably not the case. First of all, stiffness in the force enhanced state does not increase. Metabolic cost decreases, which would suggest, if anything at all, you have the same number or less cross bridges. And X-ray diffraction results do not indicate that you have a bigger mass shift of myosin towards the actin in the enhanced state compared to the non-enhanced state, the reference state. So yes, I believe in the on and off state, but I don't think it brings in more cross bridges to be attached in the enhanced state compared to the non-enhanced state, thereby explaining some of the force enhancement. I think there's a lot of evidence that that probably doesn't happen.
Okay. Arthur Cheng has two questions related to calcium. Can the calcium sensitivity of Titan be impaired by any perturbation, such as oxidative stress, inflammation, or a chronic disease state? That's the first part.
I have no idea. I don't know that. I'm really sorry, but I have never done that, and I couldn't answer that question. So okay. Sorry about that.
How about what is the myoplasmic free calcium concentration required to achieve maximal Titan stiffness? How about that part of the question?
I think the calcium binding to Titan, changes the stiffness of Titan. And we have only tested it to a PCA level of 4.0, 4.2. That's usually what we use for maximal activation of skin fibers or or myofibrils. And so we have never really gone beyond and tested whether or not it might get stiffer or not. But when we go from 4.0 PCA to lower concentrations, we don't really see a difference in the stiffness of Titan, but we have never gone higher. So I wouldn't be able to answer that question.
Okay, yeah, that's okay. We'll just take two or three more here. Wolfgang Sibo. Thank you, Walter. You showed on a slide that the marker on Titan first stretches, then maintains its position, indicating a delayed increase in Titan stiffness. Would this mean that there's no force increasing Titan support for short stretches? Greetings from Germany.
Yep. No, I know Wolfgang really well. Thank you for the question. Wolfgang, this is really an interpretation question. So the first interpretation was that we thought maybe Titan might be binding at the distal end of the segment, and therefore you would not get any marker movement. And if that indeed was the case, and you have some initial stretch, yes, that would mean that interpretation then would mean that you don't get any residual force enhancement effect for very short stretches. However, if the other interpretation is correct and you have the binding of the titan but more proximal to the marker, then all that we are seeing is an elongation of the titan segment between where it binds and the marker. And experiments that we want to do this summer with a student is that we want to place the marker at very distinct different places. And if the second idea is correct, then we should see more or less elongation of that initial segment, depending on where we place the marker relative to the binding site. And if that is the case, that we do 3 different markers and we see 3 different distances that are consistent with that, then we should be able to pinpoint exactly where that binding occurs. But the idea still being that titan is binding upon activation and therefore makes it stiffer in the active compared to the passive state, and therefore you would see force enhancement even probably in a short stretch.
Okay. Amir Hossein Ziayan asks, is there not any calcium aroused in a passive stretch? Does accumulation of phosphate affect titan stiffness?
We have not tested phosphate, and yes, even in a passive muscle, there is a certain amount of calcium there. But the argument would be it's probably not in sufficient concentrations to really significantly change the titan stiffness, or it has a certain stiffness with few bound calcium that's about the same, but is then increasing when you have more calcium. And then the last question there, I just missed that.
Sorry. Any calcium go up? Does calcium go up when you passively stretch the muscle? Not really.
Yeah. No, the other thing that I wanted to say is we have done a couple of experiments with lowering the pH. So not really looking at inorganic phosphate or a calcium, but just lowering the pH. And when we do that, then it appears at the moment, but these are very, very preliminary results, it appears that with a lowering of the pH, titan stiffness seems to increase. But don't ask me why, but that seems to be the observation.
Okay. Hetalad asks, when you eliminated the N2A region of titan in your mouse studies, did you see an upregulation of titan binding proteins, particularly actin, in some sort of compensatory effect?
We did not look for that. And so intuitively, I would think that that's probably not going to happen because actin and myosin, have a very firm structure and established structure in vertebrate and invertebrate muscle. Well, invertebrate muscles for sure. Yeah. And I'd be very, very surprised if that would change because you do something to titan. Maybe one result that I can add there is that we looked at muscles, and I mentioned, I think, David, I mentioned that to you before the seminar. We looked at muscles from children with spastic cerebral palsy, and their myosin and actin does not seem to be affected at all, but the number of, the amount of titan seems to be reduced by about half. So instead of 100% titan, about 6 titans per half myosin, there only seem to be about 3 of them there, but we did not see any changes in actin and myosin in this particular preparation.
Okay, yeah. One more question. Dr. Janessa Drake asks, a wonderful talk and discussion. Building on a previous question relating to the protection of titan on the integrity of the sarcomere, can you speak to the possibility of targeting titan for therapeutic intervention or as a biomarker for the integrity of the sarcomere and of muscle as a whole? I'm not sure what to say here, but one thing that I can say is that if we have eliminated titan in a dose-dependent manner in myofibrils and in skin fibers, and when you do that, because you have less titan, you get less passive force. But if you do that, you also get less active force.
And there seems to be a very linear relationship between the cleaved titan that we eliminated from functionally being present to the active force production. And so I'm very firm believer that for the integrity, not only the structural integrity, but also for the integrity of maximum force production, titan is absolutely essential. And some of you might know that people have tried to raise mice, deplete of a titan knockout mouse, and it's not viable. And when we take all the titan out of a myofibril and we try to activate it, we get a little force spurt, and then it immediately disappears, and you get a big mess, and you have something in front of your microscope, and you would never ever recognize it as a myofibril. So it appears that it's crucially important, not only for kind of building the structure and holding the structure together, but when you activate a muscle, also for active force production, it seems to be very important that your titan is there in the right amount.
Yeah. Let's end it there. Dr. Herzog, you have set the record for Q&A questions. I got to tell you, in our seminar series, it's almost 4:15. We're way over time. Thank you, folks. Walter, stay with me, please. Thank you.
Sure, sounds good. Thanks, David. Yeah, thanks, everybody.
Thank you for joining today. We'll see you at the next MHRC seminar. Our seminar is now over. Stay with me.